Bismillahirrahmanirrahim. Our last learning outcome from unit number 4, cell structure and function, is to describe the structure, chemical composition, and function of chromosomes. So let's start with the proceedings. So we are supposed to describe the structure, chemical composition, and function of chromosomes. So there is a sort of introduction, brief introduction. Chromosomes are thread-like structures found in nucleus of all the eukaryotic cells. They become highly visible under microscope during cell division due to extensive condensation and coiling. So chromosomes carry the heredity material that determine an organism's traits, development, and proper functioning in all activities. So Walter Fleming was the first person, and he was a German embryologist, first observed chromosomes in 1882 while he was studying dividing cells of salamander larvae. Since then, chromosomes have been identified as universal structure present in all eukaryotic organisms. So different organisms, they are having different number of chromosomes, and obviously their nature, chemical nature, properties, all the things are different. For example, in humans, 46 chromosome, mosquito, 6 chromosome, and rest of the organism, they have their different chromosomes. So let me to emphasize the penicillium, which is a fungus. It is having only one pair of chromosome. What does that mean? There is only one circular DNA in it. In that case, the organism is haploid or monoploid, we can say. Better is to say monoploid. So certain ferns, they may have 500 pairs of chromosomes. So the number of chromosomes is not depends upon the complexity of the organisms. It is just the reflection of number of chromosomes. So it doesn't ensure the complex nature. So what is the detailed structure of chromosome? Each chromosome consists of following structural components. For example, if we start with this uh, single chromosome before start of cell division, before its duplication. So it's a chromosome, chromatid arm, two arms. So this is the centromere. Centromere is also named as primary constriction because at this level, the chromosome is having a little bit of pinched off uh, appearance. So this structure is having kinetochore, different sort of complex proteins responsible for the attachment of spindle fibers. So centromere is the region, and primary constriction being the name is due to the pinched appearance. Then comes the secondary constriction, which is not present in all type of chromosome. It is present in some. So because this is a generalized chromosome, why not to mention it? So this secondary constriction is having nuclear organizer, and this area is nucleolus because there are certain genes whose function is to synthesize the ribosomal RNA. And ribosomal RNA formation is the 80% RNA formation out of 100% RNAs. So what is chromatid? Chromatid is a replicated chromosome, consists of two identical halves. They are named as sister chromatids joined together, a structure named as centromere. Sister chromatids are separated during cell division, ensuring the equal genetic distribution to daughter cells in case of mitosis. And in case of even meiosis too, they get separated from one another. Centromere primary constriction. The centromere is a specialized constricted region on the chromosome where sister chromatids remain attached. It plays a crucial role in chromosome movement during mitosis and meiosis by attaching spindle fiber via kinetochore, structure present on chromosome. So depending on the centromeric location, chromosomes are being classified into four categories. And what are these? The number one is metacentric. when the centromere is in the center and dividing the chromatids into almost equal parts. And then comes the submetacentric, when the one side of the chromosome, the chromatids are longer and other small, shorter as compared to the other one. And the acrocentric, when the distance of chromatids, the long arm is very long and short arm is a little bit more shorter but visible. And in case of telocentric, this structure is the centromere is at one end and dividing the one chromatid is attached at one end and other is at end, and the centromeric position is central one. So these are different type of chromosomes as per the centromeric position. So it is important to mention that telocentric chromosome is not present in humans, maybe some other animals and plants as well. So during anaphase, what will happen? Because chromosomes, they have to move to one pole and the other pole. So they attain a sort of shapes, different shapes. In case of metacentric, the shape will be of V type because at the center of the chromosome, the spindle fibers are attached. So the attainment of shape is V shape. And in case of submetacentric, it will be J shape. And in case of telocentric, the position will be rod shape, yeah, I shape. So same is a little bit in case of telocentric. So secondary constriction are nucleolar organizer region. So apart from the centromere, some chromosomes have secondary constriction known as NORs, nuclear organizing or organizer regions. So these regions are associated with formation of nuclei, and that region is having genes responsible for ribosomal RNA synthesis. Then comes the telomere. These are the extreme ends of the chromosomes, and they are capped with repetitive DNA sequences, and those areas are telomeres, and they protect the chromosome ends from degradation and prevent them from fusing with other chromosomes. So, telomere, they also play a significant role in aging and cellular lifespan because at every cell division they become get smaller and smaller, and there will be once again regaining of telomeric region. But in case of aged individual and during aging process, this repair will become lesser and lesser. So there is one more major concept, the karyotype. The complete set of chromosome in an organism is called karyotype. Karyotype varies significantly between species and sometimes between individuals of the same species due to mutation or chromosomal abnormalities. Now, for example, in case of Klinefelter Turner syndrome, the karyotype of the patient will be different from normal individuals. So what is chemical composition of chromosomes? So chromosome consists of three major biochemical components: DNA, deoxyribonucleic acid. DNA is the primary genetic material in chromosome. A typical human chromosome contains around 140 million nucleotides. If stretched out, DNA in a single human chromosome would measure roughly around 5 centimeter in length. The entire human genome, consisting of all the chromosomes, spans approximately 3 billion base pairs. And these all the base pairs have been sequenced through the Human Genome Project in 2001. So the proteins, approximately 60% of the chromosomal mass is protein, and mainly histone proteins. They are small and basic in properties because they are rich in lysine and arginine amino acids. Histones are positively charged, which are very rare in proteins category. But this positive charge nature is very important because DNA is negatively charged, so there will be force of attraction between them. DNA wound around histone octameres to form a nucleosome resembling beads on a string. Each nucleosome consists of 140, more or less 146 base pairs of DNA wrapped around core of 8 histone proteins, which is octamere. So further coiling of nucleosome results in 30 nanometer fiber, and then there are some other higher order supercoiling, and there will be massive compaction of DNA in chromosomes, and that compaction is very important for the sake of distribution, for the sake of equal distribution of chromosome during mitosis and meiosis. There are some non-histone proteins. Their function is to regulate the DNA replication, repair mechanism, recombination, gene expression, and maintaining the overall chromosome architecture. So then RNA, small amount of RNA associated with chromosome, and obviously the chromosome function is to synthesize RNA. So all the time transcription, when the chromatin material is present, when chromosomes are not in proper shape, not attaining or having their shape, so then the transcription is being carried out by the metabolic, by the machinery of the nucleus. So organization of chromatin, the substances making up chromosomes exist in two states. So chromatin is a prior stage of the chromosome. When the chromosomes, when the cells, they are not in dividing phase, then their chromosome, they attain, yeah, they may have the chromatin material. And chromatin material is uncondensed, uncoiled form of chromosome, which is actively involved in its function. So, two types of chromatin material. One is euchromatin, perfect chromatin performing the active stage, and the function of DNA is being carried out. Heterochromatin, highly condensed chromatin, and genetically inactive because it will not transcribe into messenger, into RNA, any type of. And there are two types of heterochromatin. Constitutive heterochromatin always condense centromeric region, for example, centrometric meric region. And then the facultative heterochromatin may become uh, a euchromatin under certain conditions, for example, inactive X chromosome, and in one female, one chromosome may be inactive, in the other female, maybe the other chromosome may become active. So what are the functions of chromosome? Last part we are supposed to provide in our syllabus. We are supposed to answer. Chromosome performs several essential biological roles: storage of genetic information. Chromosome carry genes encoding proteins and RNAs critical for organisms' development, metabolism, physiology, I mean all life activities. Transmission of heredity information. Now, this is important. To carry genetic information is one thing, and to transmit it, the genetic information to other cells is another important task. During cell division, mitosis or meiosis, chromosome ensure equal and accurate distribution of genetic material to daughter cells, which may be gamete, may be somatic cells, and this will preserve the genetic continuity across generation if the cells and the under consideration cells are gametes. Gene expression. So structural organization of chromatin influence gene accessibility, and euchromatin allow gene expression, heterochromatin silencing those genes in which are under the heterochromatin region. And maintenance of genetic stability. Telomeres protect the chromosomes ends, and DNA repair protein associated with chromatin help to preserve the genetic integrity. So determination of sex, for example, X and Y chromosomes in human and other animals. So their function is to determine the sex of the individual. So role of evolution, this is important. Chromosomal mutation, duplication, deletion, inversion, translocation, they contribute towards the genetic variation, and genetic variations are the cause of evolution. Importance of chromosomal integrity. So loss of chromosome, monosomy, or presence of extra chromosome, trisomy leads to genetic disorder, which are quite familiar to us. Down syndrome, the trisomy of 20, chromosome number 120, chromosome number 21. There are three copies of it. Turner syndrome, monosomy, X chromosome, one will be lost, and Klinefelter syndrome, in which XS are in double form and Y is single. So hence, maintaining chromosomal integrity is vital and normal function of the chromosome to maintain the health. What is the conclusion? Chromosomes are marvels of biological engineering, packaging enormous length of DNA into microscopic spaces while precisely controlling gene activity and transmission of hereditary traits. So their structure and function are fundamental to life, making them central to understanding genetics, cell biology, development, evolution, and onset of certain diseases if they are genetic in nature. So that is all about the chromosome structure, function, and chemical nature. So this is end of unit number 4.